Dr. Hansen here, and today I want to look at lipids. And as a physician, you are going to order lipid panel blood studies. That'll be one of the most common tests you order, especially during physicals. What does a lipid panel measure? And I'm not interested in every detail. When I ask questions like this, I mean, what are the main things you think a lipid panel would test for? What do you want to know? And the answer is simple. It measures many things, but it especially measures cholesterol and triglycerides. Why do you measure these concentrations? Please pause and answer. Because most of your patients are going to die from cardiovascular disease. Coronary artery disease results from a buildup of fat in the lining of your arteries. And in fact, when you eat a big Thanksgiving dinner, your blood plasma changes in appearance. Can you see the difference between these two tubes of blood plasma? Please tell me what the difference is and explain it. I was really impressed when I first saw this figure. Before eating a large meal, your blood plasma looks relatively clear. After the large meal, it's very cloudy. What do you think is making it cloudy? Well, it's lipids, but it's not just cholesterol floating around. It's not just triacylglycerols floating around, because those molecules are too small to interact with a relatively large wavelength of light. What is the cloudiness caused by? It's caused by lipoprotein, huge spherical transport particles that will move cholesterol and other lipids through your blood. Lipoproteins have a triacylglycerol and cholesterol ester core surrounded by a monolayer of mostly phospholipids and some cholesterol, along with apolipoproteins. We will talk a lot about good and bad lipoproteins when we get to cardiovascular. But right now I want to point out that fat is evil. As I said, most of your patients are going to die from coronary artery disease, and one of the major modifiable risk factors for cardiovascular disease is hypercholesterolemia. For that matter, I personally am taking statins right now. At the same time, let me point out that fat is good. In fact, just as you can have an excess of harmful fats, you can have a deficiency of good fats. And there are many different types of good fats. This is especially important during fetal development. It's important in early childhood and even in later life. Deficiencies can contribute to cancer, Alzheimer's, diabetes, schizophrenia, and autoimmunity. In fact, I'll wager that a number of you are currently taking fish oil supplements. What is it that you get in them? Please pause and tell me. How about omega-3 fatty acids? And just as omega-3 fatty acids are important, also omega-6 fatty acids are important, and we'll look into that in more detail. In addition to the good essential fatty acids we absolutely need, it turns out that even the fats we call bad are only bad currently because we take them in excess. Triacylglycerol and cholesterols are profoundly good and helpful and needed in the correct context. In fact, triacylglycerols form the largest reserve of energy we can store in fat. This is what enabled prehistoric man to survive thousands of years through cycles of constant starvation, alternating with feasting as they might trap a huge animal. And even currently, we are constantly storing and drawing upon our fat as a fundamental part of our daily metabolism. Cholesterol is an essential component of every membrane in your body. It's very important in myelin to stabilize our nervous system. It's also the precursor used to make bile detergents, steroids, and vitamin D. Dietary fats are important in helping us take in fat-soluble vitamins. Omega-3 and omega-6 polyunsaturated fatty acids, as I mentioned, are important to regulating the immune system, and they're also very important for development of the brain during fetal years, early childhood, and throughout life. I want to make a big point that the highest concentration of lipids in your body, apart from fat storage in adipose cells itself, is in your brain. And in fact, I think it would be quite fair if I call you a bunch of fatheads, because the concentration of fat in your brain is extremely high. Furthermore, the types of fats in your brain are unusual. There's more cholesterol in your brain, and there's more polyunsaturated fatty acids there than in most other tissues. The brain is a very special type of fat. Review learning objectives. Hopefully you understand the system for numbering carbons using Arabic numeral or Greek numbers, so you can just apply that again here, my next talk, the one after that. Likewise, organic nomenclature. Hydroxyls, carboxylic acid, or ester bonds are going to be used every time. And for that matter, an awful lot of lipids are linked together through an ester link. And the one mechanism, a nucleophilic attack, can be applied the same as always in order to hydrolyze that bond. And again, you learn one mechanism, apply it over and over and over. You have less to learn. What is the most prevalent form of lipid that's in your body right now? What's the most prevalent form of dietary lipid? And for that matter, what fraction of the lipid in your body or in your food do you think is composed of that one type of lipid? Please pause and answer. Okay, I hope you've done that. It's real important to pause and answer these questions to get a feel for where you're at. I do think quite a few of you probably know that triacylglycerols are by far the most prevalent form of lipid. In fact, about 90% of the lipid in your body is triacylglycerols. They're extremely prevalent. For that matter, butter, olive oil, bacon grease, they're comprised almost completely of triacylglycerols. There might be some water mixed in, but 99% of the lipid in these greases and oils is triacylglycerol. Can you interpret the name triacylglycerol? Well, please pause and do so, but I'm going to move on. Of course, tri as in three. Acyl is an extremely important word that you should know. Acyl means that it is an ester link to a fatty acid. So there's three ester links to fatty acids. Glycerol, well, all means it's an alcohol. And glycerol literally comes from glycos, from sweet, but this glycerol, this sweet substance, is a 3-carbon alcohol, glycerol. You absolutely need to break all of these things apart. We're going to talk about many forms of glycerol over the next 2 years. It's in your interest to know exactly what it is. And here we're showing the 3 ester bonds that link them together. Whenever you're thinking about complicated chemical compounds, whether it's your genome, your proteins, oligosaccharides, polysaccharides, or these lipids, such as triacylglycerols, it really helps a lot if you look at the individual building blocks. Every continuous carbon chain represents a building block. In this case, a fatty acid, a fatty acid, a fatty acid. And then where you see the oxygens interrupting the carbon chain is where you have a different building block, in this case the glycerol. Very important to think that way. Now my question is, of these 2 extremely greasy molecules with all these hydrocarbons, which of these 2 represents olive oil and which of them is more indicative of butter? Please pause and answer. It's always in your interest to try. Well, what's the difference between butter and olive oil? Butter is a solid at room temperature, whereas olive oil is a liquid. Which of these will more likely be a liquid? Generally, saturated fatty acids are fatty acids where every carbon is completely saturated with hydrogen. Unsaturated means that there's at least one double bond. And in most lipids, this double bond is going to be cis. Please pause and tell me what cis means. Well, we have 2 types of bonds. Trans means if you look at a typical bond, the carbons on opposite sides of the bond are trans or across from each other. A cis double bond means that if you look at carbon atoms on both sides of the double bond, they are on the same side of the double bond versus opposite sides. So saturated fatty acids tend to be all trans in their lowest conformation, When you have a single cis double bond, you can no longer have an all-trans structure. There will be a kink. Now here I'm showing 3 fatty acids, each with a single double bond, putting a kink into them. And this is going to represent butter or olive oil. Please answer. Just as with carbohydrate polymers, if every bond can be trans, then tighter packing is allowed, and if there are cis bonds, that can put a kink. So for example, here we see all of these straight, completely saturated fatty acid chains can pack together tighter than the ones which have a forced kink in them. And therefore, these straight bonds will have a higher melting point. This corresponds to bacon grease. The ones which have a cis bond on every carbohydrate chain represents olive oil. This omega-9 fatty acid is specifically called oleic acid. In fact, in the case of olive oil, all 3 of the fatty acids attached to the central glycerol in the triacylglycerols are oleic acids, an 18 carbon chain with a cis bond on carbon 9, making this an omega-9 fatty acid. In contrast, most animal fats have a saturated fatty acid linked on carbon 1 and carbon 3 of glycerol, and the central carbon has usually an oleic acid or some other type of unusual fatty acid, maybe a polyunsaturated fatty acid. Here I'm reminding you of what I described in part 1, that completely saturated means completely saturated with hydrogen, and the tendency is for these long fatty acid chains to be mostly linear. Regardless of the nature of the various fatty acids which might be linked to a central glycerol in triacylglycerols, triacylglycerols are so greasy they tend to accumulate into huge blobs of fat. Those fats can be solubilized with detergents, which are amphipathic molecules. Common laundry detergents contain a sulfate head group linked to a hydrocarbon chain. Bile acids in your intestine contain a charged head group linked to a cholesterol that has been hydroxylated to make it one side greasy and the other side hydrophilic. The greasy parts of these amphipathic molecules can stick into the blobs of fat. The charged groups can be pulled out by water, and this can help solubilize blobs of fat. A useful but simple exercise would be to show hydrolysis of all of the ester bonds in these triacylglycerols. If you want, take a stab at that by pausing the video now, but I need to move on, and here I'm showing that lone pairs of electrons on the oxygen atom in water will nucleophilically attack the carbon in the double bond to oxygen. Electrons in that double bond will resonate up onto the oxygen to form an oxyanion, then they'll resonate back down and kick off the glycerol, such that this has been an oxidation reaction. The oxygen from each water molecule was inserted into the compound to form a carboxylic acid on the fatty acid, and thereby to kick off the hydroxyl on the glycerol. In part 1, I defined omega-3, omega-6, and omega-9 fatty acids. Can you go ahead and categorize each of these accordingly? In fact, each of the fatty acids in this figure are quite important. The nutritionists refer to linoleic acid and alpha-linolenic acid as the only 2 essential fatty acids. They are essential because humans are unable to insert double bonds near the hydrocarbon tail opposite of the carboxylic acid end of a fatty acid. In fact, the closest double bond that a human can insert is at the omega-9 position. Almost all the chemistry with fatty acids occurs at the carboxylic acid end. That's where they get linked in ester bonds to other compounds. That's where fatty acid biosynthesis occurs. You keep adding 2 carbon units from acetyl groups to elongate at the carboxylic acid end. That's where fatty acid oxidation occurs to make energy. You keep breaking off 2 carbon groups as acetyl groups linked to coenzyme A. And that's where you can insert double bonds using desaturase enzymes. The electronegativity and resonance of that carboxylic acid group makes all sorts of chemistry possible. In contrast, the hydrocarbon tail, it's hard to get a handle on that. You can't do much interesting chemistry there. Accordingly, humans can never take linoleic acid and change it from an omega-6 fatty acid to some other type, omega-3, omega-9. However, humans can alter the carboxylic acid end by elongating it with 2 carbon units and by desaturase enzymes to convert linoleic acid to arachidonic acid. And for example, humans cannot take in alpha-linolenic acid and convert it from an omega-3 to an omega-6 or omega-9. It will always be omega-3. But again, they can use desaturase enzymes and they can insert 2 carbon units to convert alpha-linolenic acid to eicosapentaenoic acid and ultimately to docosahexaenoic acid. I want to point out right away, however, that I prefer to take in some DHA and EPA from fish because the level of activity of these desaturase and elongation enzymes in humans is not completely understood. Docosahexaenoic acid, eicosapentaenoic acid are so important. For that matter, eicosanoids are important that I do require that you memorize those names or understand them. It's easier to interpret them and know a little bit of Greek than it is just to memorize them as gibberish. Not only are they involved in a whole bunch of experiments looking at everything from dementia to heart disease to diabetes to metabolic syndrome, but a lot of your patients are going to be taking EPA and DHA, and these may end up doing weird things to their immune systems. An even more important learning objective is to differentiate functionally between omega-6 and omega-3 fatty acids. It turns out that omega-6 fatty acids are overall pro-inflammatory and omega-3 fatty acids are overall anti-inflammatory. That is, arachidonic acid and eicosapentaenoic acid are each precursor for immune signaling molecules which tend to have opposite effects. The most prevalent of all forms of lipids are these triacylglycerols, which are considered simple lipids. I guess they're considered simple because, well, 90% of the lipid in your body is of that form. The only other simple lipid I'm aware of is a free fatty acid that you would get if you would hydrolyze these ester bonds. In contrast, complex lipids would pretty much include anything other than triacylglycerols and free fatty acids. For example, glycerophospholipids contain lipids and glycerol and also a phosphate and maybe other groups. One type of glycerophospholipid is phosphatidylcholine. I don't like the name because tittle must evidently mean 2 fatty acids, a glycerol, and then you have the phosphate and choline up here. I don't like the name, but phosphatidylcholine is important enough and prevalent enough, and there are critical enzymes which include phosphatidylcholine in the name, involved in diseases. I do require that you be able to know it. And since you're going to know phosphatidylcholine, how about phosphatidylinositol? Tittle again must mean 2 fatty acid linked in ester bonds to glycerol. Phosphate, we know what that is. Inositol is simply a sugar. A lot of this nomenclature you can interpret, and you have to go a little step further with some of the names. Sphingolipids is a weird name. Sphingolipids look very much like glycerophospholipids. Given the structure of a glycerophospholipid and a sphingolipid in cholesterol, I expect you to recognize and categorize them, but please don't get bogged down in the details of the differences between them. At the same time, I want to compare a sphingolipid to a glycerophospholipid to give you insight. So please listen to my detailed description and say, does this make sense? But don't try to memorize the details. The overall structure of a sphingolipid looks an awful lot like the overall structure of a glycerophospholipid, but there's a big change. It's as if you've taken the glycerol and one of the fatty acids and replaced them with a single lipid chain, so that you have a hydroxyl at the top of both. You have a hydroxyl sticking out of glycerol corresponding to a nitrogen sticking out of the sphingolipid. You have a ketone in the ester bond sticking out a glycerol phospholipid, but there's a hydroxyl here. And so instead of an ester bond, you simply have a carbon, a hydroxyl, and below the ester bond you have a double bond here, which you don't have in the glycerol phospholipid. The mimicry is pretty remarkable. And why is it called a sphingolipid? It's a real riddle because it was named after the riddle of the Sphinx. When they were looking at all these different types of lipids and different fatty acids linked to different compounds, they could not figure out what this thing is. Come on, there was no glycerol, but there was something that kind of mimicked glycerol. There was a continuous fatty acid chain. There was a double bond in it, a hydroxyl. There's an amide link rather than an ester link. Very confusing. But sphingolipids are quite important. You're going to have to know about them. And this would be called a glycolipid simply because there's a sugar linked to a lipid, much like a glycoprotein. Can you explain to me why I've drawn a line in the sand? What is the difference between the two categories of lipids I've defined with the dotted line? Please pause and answer. The answer again is structural. The lipids to the right are profoundly greasy. Cholesterol ester is the greasiest molecule in your body. The next most greasy molecule is triacylglycerol. These things are always buried in blobs of fat. In contrast, the compounds to the left here are all amphipathic. You have the very greasy, long extended fatty acids on one side. At the other end, you have a hydrophilic group. In the case of glycerol phospholipids, you have charged groups. You have huge amount of hydrogen bond potential. In the case of cholesterol, you have a simple hydroxyl. But because that's at the very end of a long molecule, absolutely, cholesterol is amphipathic. To be clear, cholesterol certainly is also hydrophobic. Being amphipathic does not rule out something being hydrophobic. Cholesterol can be categorized both ways. It's always found buried in membranes, but the hydroxyl is always trying to stick out of the membrane, making it polar. Cholesterol is amphipathic and hydrophobic. To be even more clear, cholesterol is certainly less hydrophobic than a cholesterol ester or than a triacylglycerol, and it's less amphipathic than a glycerol phospholipid. There's a whole spectrum of increments between extremely hydrophobic, extremely hydrophilic, extremely amphipathic. Accordingly, to the right, the extremely greasy molecules are for storage. The amphipathic ones are used in forming membranes. And I want to look at the character of these lipids in membranes. How flexible, how dynamic a membrane is depends upon the nature of the lipids in that membrane. Can you categorize these three lipids based upon which will lead to the most dynamic fluid membrane and which will lead to the most constrained stiff membrane? How stiff a membrane is depends on how tightly its constituents can pack. The amide bond in sphingolipids is linked to a completely saturated fatty acid, whereas glycerol phospholipids very often have an unsaturated fatty acid through their corresponding ester link. Furthermore, the central lipid of a sphingolipid, the sphingosine shown in purple, has all saturated bonds except for one double bond, which is trans. Not only does a trans bond not put a kink into one of these, it also actually stabilizes the trans conformation to make sphingosine even more straight and linear than a completely saturated fatty acid. Accordingly, sphingolipids can pack extremely tightly together in contrast to glycerol phospholipids, which usually contain a fatty acid that is unsaturated in position 2, either oleic acid or perhaps some other polyunsaturated fatty acid. Accordingly, sphingolipids can pack very tightly together, whereas the kink in the glycerol phospholipid linked at carbon 2 almost always causes the membrane to be more dynamic. How about cholesterol? Cholesterol is a brick. Look at that fused ring system. That's not going to bend and flop around anywhere. And there's another characteristic to this. Just as the sphingolipids have the very straight side chains that can pack tightly together, it turns out if you look at the conformation of cholesterol, it has a very flat side that packs very nicely against the saturated side chains of a sphingomyelin. Accordingly, it turns out that sphingolipids and cholesterol tend to group together and to aggregate into lipid rafts. Again, another learning objective that will be very important next year. And in contrast, glycerophospholipids with a kink tend to also accumulate in other regions of membranes. Based upon what I've described so far, and upon your knowledge of the brain, where do you think there will be more cholesterol, and where do you think there will be more polyunsaturated fatty acids within the brain? Will there be more in white matter or more in gray matter? Please pause a minute and think about it. A lot of you should be able to come up with this answer. Don't get bogged down. Okay, what is the difference between white matter and gray matter? Generally, the white matter contains those very long connections, the axons. In contrast, the gray matter is the cell bodies and the synapses. And what causes the white color? Well, those long axons need to be protected. They need a layer of insulation, which you call what? Myelin. Myelin contains a large amount of cholesterol because it's so stiff and it's so protective. In contrast, in the gray matter, you need polyunsaturated fatty acids. Why? Please pause and answer. The synapses have to have very dynamic membranes. The vesicles containing the neurotransmitters very rapidly need to merge with the synaptic membrane to release those neurotransmitters repeatedly, rapidly, and in quick succession. So we want very dynamic membranes which have very flexible polyunsaturated fatty acids. The concentration of polyunsaturated fatty acids in the synapses of your brain are higher than anywhere else in your body. I'm hoping that this background will help you when you hear my presentations. And in the meantime, don't hesitate to stop by if you have any questions. Thank you.